Yeah, well, welcome back, everybody, and pass the chair to lead us off.
Okay, well, the agenda has a review of day one and the discussion of recommendations from day one. I'd rather just put that off till later. We'll just get started while we're fresh. So we jump into the biology program.
Thanks very much.
So Seth, take it away.
Yeah, can I be made a presenter, please? Okay. Well, good morning, everybody. Happy to present the report on current and future biological system science research activities. We're going to provide some updates on three particular areas. That is migration population dynamics, specifically answering to some of the requests and recommendations from SRB025. Then we're going to be providing some new data on the maturity schedules by Colin. And then later on, we're going to finish with the last two projects, UMR, again, responding to some of the requests and recommendations from SRB024 and a brief update on current status of the work on the tax protection. So we're going to start with the first research area, that's migration and population dynamics. And Andy Jason Weekend is going to speak to that. So you can pass the slides.
Okay, thanks, Josette. So, yeah, so I have a couple slides here. I put together a few slides to serve as an update on some results that were generated as a result of a recommendation made at SRB024. And before getting into that, I just wanted to start with a little bit of background information just to reorient everybody to the study design. So the current objective of the population genomics work here at IPHC is really aimed at resolving the genetic structure of Pacific halibut in IPHC convention waters. And a very key important aspect of this study is that we're exclusively utilizing samples collected from spawning groups. And that essentially means these fish were collected during the wintertime when fish are assumed to be on the spawning grounds, and therefore a representation of spawning groups and the geographic areas we sampled, which you can see in the map here. We have samples from British Columbia, Central Gulf of Alaska, Bering Sea, Central Aleutian Islands, and Western Aleutian Islands. And these samples were collected over a fairly broad time frame between the years of 1999 and 2020. And the method that we're utilizing to generate the raw data for this project is called low coverage whole genome resequencing. And essentially this method is a very high resolution technique that has a capability of sampling every base pair in an individual's genome. So in the box on the right, I have some metrics related to the processing of the raw sequence data into the data set that we're utilizing for analysis. But that has been presented before, so I'm not going to go into any details there. However, the analyses that we've conducted so far have not been able to identify any discrete genetic groupings of Pacific halibut residing in IPHC convention waters. And essentially what that means is that we're not able to provide a genetic basis or justification for recognizing distinct units of the stock for assessment purposes based off the analysis of this data. So at the previous SRB meeting, the SRB recommended that the Secretariat test for stock structure using only male Pacific halibut. And if I recall correctly, this was made after some discussion of tagging results that seem to suggest that male Pacific halibut may not move as far between their, the location in which they were released and the location in which the tag was recovered. So essentially, to address this, we conducted principal components analysis, followed up by k-means clustering separately for each sex, and this was using the same procedure that we used to analyze the entire data set. That is, both sexes together. And the basic procedure here is first estimate a covariance matrix directly from the genotype likelihoods that we've estimated from our data. Prior to that, we filter out any SNPs that have a minor allele frequency of less than 0.05. And then following this, we perform eigen decomposition. That's done in R. And then following that, we then perform k-means clustering, and that's also in R. In this, we tested a range of k values between 1 and 20. And k is essentially the number of clusters you're attempting to partition your data set into. So here we have the results of the various PCAs that were conducted. And so starting on the left, we have the PCA where both sexes are analyzed together. And this has been presented before, but I included it here for purposes of comparison. And so again, in these figures, each point represents a single individual, and each individual is colored by the geographic area in which they were sampled in. And then for these figures, males are represented by triangles and females are represented by circles. And the lines represent 95% confidence ellipses for each geographic area. Okay, so moving to the right, we have the female only PCA. And you'll notice here again that we're not actually forming any distinct clusters of individuals with this analysis. Then moving one further to the right, we have the male only PCA. And again, we're not detecting any distinct groups with this analysis, and specifically groups that might relate to spatial structure. If you look at the confidence ellipses in this male only PCA and kind of the relative position to one another, they are very, very similar, if not track the analysis where both sexes are analyzed together. So I guess the point I'm trying to make here is that by focusing on each sex independently, it's not like we're gaining any additional ability or power to identify discrete genetic groups here. So I guess to kind of move beyond the method of the eyeball to something that's a little more objective. These figures here are two different model support metrics that we use to evaluate the clustering solutions for our k-means clustering analysis. So on the y-axis, we have total within cluster sum of squares, or BIC, and on the x-axis, we have the various values of k tested. And the blue points and lines are associated with the female-only data set, and the yellow points and lines are associated with the male-only data set. And the fairly constant and continual decay that we're observing as we attempt to partition that data set into higher numbers of clusters makes it very difficult to confidently identify a single value of k that might best describe our data set. And oftentimes when you see these sorts of patterns, it's likely the case that a single cluster best describes the data. So in summary, we failed to detect discrete genetic groups of Pacific halibut when each sex was analyzed independent of one another. And this is similar to the previous results of the analyses that we've presented. Again, probably owing to really a considerable degree of gene flow among areas. And yeah, that's likely due to migratory behavior of the species. So that's all I have for the genomics. So I guess if there's questions, we can do those now, or pass it back to Joseph. Yeah.
So the section is migration, population dynamics. I'm just wondering, is there more coming on migration, on movement?
Not at the moment.
Okay,
No,
so
I'm not contemplating doing any specific work on that.
because I mean, if there's no inherent population structure, which is what it is. It's all great work and highly defensible, and that's great. Movement seems to be the potentially the most impactful thing that remains highly uncertain in the whole situation here. Are there any plans? I can imagine it would take a lot of effort. I know it's been tried before. Genetic ways of gene tagging is used in some places, which is not as complicated as CKMR, but maybe there's other, what do you think about that?
Well, I mean, so some of that, if you recall, so at the last meeting, we had some results of essentially assignment testing. So you're essentially utilizing an individual's genotype to assign them back to a reference population, or based on the allele frequencies within a reference population. And we had really no ability to actually, like, the assignments were, I think the correct assignment rate was like 36%, and that's because all the fish were being assigned back to the Gulf of Alaska. So I mean, that's like, I mean, I guess that's so with like, this sort of stuff, the genetic work, I mean, those kind of, can provide like indirect measures of movement, right? And then like the assignment testing would be, a tool that you could use to kind of leverage the genomic data to get more direct measures of movement. But yeah, the resolution among, the spatial areas is just not there in order to kind of go from to take it to a more like direct measure of migration. And then, yeah, I mean, you basically have to go up the rungs of the ladder to move to something like a CKFR or kind of maybe kinship-based sort of approach.
Or like gene tagging is essentially just using an individual DNA fingerprint, but I imagine that probably is pretty, you need to sample a lot.
Right.
Different captures, because
Right.
you're talking about essentially individual.
Right.
Tag
Yeah,
captures.
exactly. So yeah, I mean, you'd be talking from moving from like a population-based approach to kind of a more individual thing.
Based on kinship relationships, identified genetically.
That's not what I'm saying. It's not what I'm suggesting. What you're saying is that won't work.
I mean, based on this, it doesn't seem like that would be the case, but
I think this results sort of force us to go back to the drawing room and try to revisit the topic of migration.
That's what I'm saying.
In other manners.
Yeah. Is it time?
Because we're putting some expectations into our ability to assign individuals back to the population of origin and then infer migration that way. But from the end, it just found out that our ability to do that is not present. So yeah, we're gonna have to re-examine future work on that.
Do you still tag fish like traditional fly tagging?
We tag all the sublegal fish on the survey that we not sampled. But we don't get a huge number. I think our recovery rate is less than 10% on those fish.
Around 10%.
Is the reporting rate? Your estimates of reporting rate?
Not current estimates. We have had reporting rate estimates. We provide a hat or $10, I think. And it's a free fish. So a tagged fish with an IPHC tag can be kept by anybody under any circumstances. It's free. It doesn't come off the quota. It can be kept by trawlers, anybody, recreational fishermen. So there's pretty good incentive. I think we would expect reporting isn't bad. It's not like some programs where you talk to someone and they have a whole drawer full of tags because they like to collect them. I think we get them back when people recover them.
What do you mean recovery is 10%? Their life, like expected lifetime?
Yeah, we generally get less than 10% of our tags back. I think the smaller the fish we tag, the lower that rate is. The larger they are, the higher. We always get a few within a couple of months of releasing them, and then it goes down substantially after that. But yeah, over their lifespan, we just get less than 10% of these smaller fish back.
Tagging over the head, are
We're not. We've done a lot of different kinds of tagging. I think that the opercular, sorry, we're not doing opercular tags on the sublegals, are we? We're doing the,
No,
yeah, we are.
It's a
Yeah, we are.
It's a record.
So they put them through the, right.
Retention's good.
It seems to be. We've certainly got some long-term recoveries with that method, but Tim did holding studies with a number of different tagging methods years ago. Found that like internal tags get digested, pass through the abdominal wall. They're like catfish in terms of their ability to get rid of internal tags. And our satellite tags all go through the
Trachea forest.
yeah.
I wonder if you thought experiment or maybe someone can discuss later, like if you knew movement rates perfectly, how much would that improve performance? And then our discussion of whether the MSE or, whether a management procedure is ever going to be in the cards for this fishery, whether it's even possible to measure. But, so is migration worth? No.
There's a comment from Kurt online saying he took 2 calls from sport fishermen this year who were interested in the tags. One of whom released the tag fish, but kept the tag and jotted down the tag number. But they seem to still generate interest in people. Just anecdotal.
We're also working on a new portal for tag recovery, so I'm hoping to like make it a little bit more attractive for people to report the tag. So the new portal will be like you put the number and you can actually find out where the fish was released, all the details. So we hope this will also attract people to. So this is something like maybe a couple of weeks away, and we'll be pushing that. So it's going to be a website where you can put the tag number, you can see all the details, and you can directly report to us that tag by clicking instead of like finding somebody in the port, so like one of that makes you it's in the port calling or something. So this should be hopefully improving the whole process, make it more just a general like more appeal to actually use that. And that will be also part of that. So we have also some of the other mark of tail pattern recognition project. So this will also allow people to directly just to take a picture on the phone. This will help ability to collect.
But I think generally just this genetic part of these studies should be published in a scientific journal, because it's the trend is to try to detect this kind of substructuring of big fish stocks, and people tend to think that it exists, and actually showing evidence that it's not always the case would be really important.
Oh, yeah. Yeah. I mean, we definitely intend to, publish this. So, like next year sometime, obviously.
Yeah, in 2025, we can issue impact. Andy is processing some additional samples that were not included in this initial analysis. In 2025, we'll have manuscript ready.
Yeah.
I don't know if for the SRB for June, but definitely by the September meeting, we should have a draft.
It seems to be a pretty straightforward story. And it could be maybe more powerful as a small, like a letter or short communication, like constellation letters or fisheries go to paper.
Yeah. I mean, I guess, we haven't got to the point of like, picking out journals yet, but, but I think, like with this kind of thing, you do have to kind of defend it, so there's a lot of, essentially what we're trying with at least the previous SRB, multiple methods kind of come to the same conclusion. Kind of like, I guess it requires a little more defense than very clear, nice results. You know, is what I'm saying. Not that these aren't, but you know, you kind of really have to show that. So, at least that's sort of how I see that. But
To your question, Sean, I think particularly with regard to 4B, now that we've identified that there is a unique genetic component that we need to conserve for its own value, the question really is, in terms of optimal management, what kinds of harvest rates are appropriate for that area? Is it getting seeded by fish from other areas? And what we really need to understand is the connectivity that's creating the lack of genetic signal associated with the larval and egg and larval phase? Is it associated with dispersing juveniles? Is it actually adults moving back and forth? And we've done, Joseph's team has published on oceanographic modeling of larvae to try and understand that dispersal, and now on movement of juveniles inferred from patterns in the trawl data. But we still, for 4B, I think we still don't really understand which life history stages are well connected across that boundary. And you know, I think Andy's done a good job of describing that just because we don't see a genetic signal doesn't mean it's just panmixia. I mean, we could have actually very low rates of exchange and still not get a genetic signal. I mean, we could have our harvest rate in 4B could be way higher or lower than it should be, depending on, for optimal yield, depending on the type of linkage that's going on there. At the extreme, it's a sink population where recruits end up there occasionally from the rest of the stock, but adults don't really move. And then, you know, I think the conclusion is you should just catch them when they're there and then wait for the next batch to end up there. On the flip side, if it's a mostly self-sustaining population that just has, you know, a few fish moving back and forth, then we'd probably want to custom tailor harvest rates to that area to maintain that stock. One of the avenues we've been working on is trying to understand how much habitat there is for post-settlement juveniles in each of our regulatory areas to be able to understand what level of productivity could be produced endemic to each area, to give us an idea of whether that's consistent with the yields that we've seen taken out of there. Basically, can they pay their own check, or do they have to be subsidized by individuals from outside the area to just give us a sense what the gross surplus production might be?
Do you think that's the bottleneck?
Well, we don't really know. I mean, we see there's been a lot of sampling of juvenile flatfish, and halibut show up in fairly specific places, and they're not present in a lot of places that seem like they'd be great. For example, the west coast of the U.S. has a lot of shallow sandy habitat, which in Alaska, shallow sandy habitat's great for juvenile halibut, but there apparently are no juvenile halibut on the west coast of the U.S., despite having ocean beaches all the way up. So that's one avenue we've been working on, but we haven't really made a lot of progress on.
What about parasite genetics? I've seen a couple papers suggested that the genetics of parasites were has a better stock structure. Themselves.
it's a really interesting idea. But yeah, I haven't seen any. I guess I'm not familiar. So if you do have literature, I would say if you don't mind,
Pass along.
that would be great. Yeah.
I mean, the idea is basically that the parasites are
Right.
genetically distinct. And
Yeah.
if you've got parasites from that area, you know that they pass through it.
Yeah. I guess, right. You're more kind of relying on the life history of the parasite rather than, Interesting. Yeah.
That's long ago. They looked at parasite loading.
Right.
Right.
Yeah, this is like the next phase of this. Parasites often have more stock structure than fish.
Yeah. I mean, yeah.
So there is a master's thesis on parasite loading in Pacific halibut, but the thought and it didn't find a whole lot of structure, but the genetics concept is an intriguing one. Sure.
Yeah, because you could find the same parasite
Right.
loads.
Yeah.
Stock structure places.
Unfortunately, the type of samples that we've used for this analysis, sort of fin click. So parasites are usually located in liver, and muscle, and Pacific halibut. If we had sequenced probably muscle or liver, maybe we would have picked up some parasite sequences that we would be able to probably identify species. But
There's certainly external parasites too that we could pick off of different halibut in different areas that are common species throughout the range, but they may have different genetic signatures. So
yeah, it's interesting. Yeah, it would be interesting to read all.
Interesting. Yeah, really.
Okay, thank you. That's great.
All right, so we'll move on to the next area. So Colin Jones will speak to some of the recent data that he's generated for summer, and he just presented at the American Fisheries Society last week. So,
All right, so lots of updates since June, so I'm excited to present the results here. So just a quick overview. So starting in 2022, we set out to collect histological samples for maturity on the Fishery Independent Subline Survey. And we continued that in 2023, and we also did it this year, which I'll talk about at the end. But this map just shows kind of a geographical distribution of sample collection. And note again that the FIS was reduced in 2023 and only covered regions 2 and 3. So from a temporal component, we'll only be able to compare those two regions and not the two regions out west. And we've collected over 2,000 samples between these 2 years. So these next two slides are the figures that I had presented at SRB025 in June, just to give a background information. So we were comparing GLMs and GAMs, and with a log transformation of age by region. And so this is the output for the GLM, and what you see are the four plots by region. And the data on these plots for the model outputs, the data is binned. And then the dashed lines that you see indicate age at the proportion of age at 5%, 50%, and 95%, just to kind of give you an idea, kind of gives you a visual estimate between the regions. And what we found from the GLM was that it is showing a higher proportion of maturity at the younger ages. It doesn't quite encapsulate the steepness of the ogive curves at the very young ages, usually between ages 5 and 7. And then in comparison, here was the GAM output. You can see it encompasses those younger ages a lot better, but also tends to truncate the curve towards the older ages, which we are seeing through histology that there are some females that are older than age 20 that are immature. And you can see that the plots using the GAM definitely show difference in nonlinearity, in particular region 3, which had effective degrees of freedom of 5. And I wanted to point that out because you'll see when we incorporate a year effect into the model, it greatly impacts the nonlinearity of that curve in particular. So then to show the 2023 data, first I wanted to just show everyone the raw data for biological regions 2 and 3. And so in these plots, each bubble represents a specific age, so that the data is not binned here. And you can see that in comparison to 2022, there's two distinct things. It looks like there's a lot higher proportion of mature females in region 2 for 2023 in comparison to 2022. But then in region 3, it tends to show a similar trend. So then now requested back in June, I'm gonna first show just the 2023 data modeled separately, and then I'll show some model comparisons of the year effect models. So this is the GLM for 2023 for regions 2 and regions 3. And it shows similar model trends to 2022 in that you see the ages 5, 6, and 7 are showing a higher proportion mature in comparison to the GAM. And so here's the GAM output for 2023. Region 3 is showing a lower value of effective degrees of freedom compared to 2022. But also region 2 is showing a higher, basically equal nonlinearity for 2023. But now we can get into the encompassing a year effect model. So Ray and I did a bunch of model comparisons for the GLM as well as the GAM. And we found that for the GLM, the addition of a year region effect was the best fit model. And then for the GAM, we did 2 different model comparison. One was with a second smoother function and one without. And what we found was that the ASC values were identical. So we chose to use the only one smoother function model with the addition of a year region effect. And that closely represents also the GLM to do a comparison between the two. And so these are the model outputs with the year effect in the model. So what you see here, first for the GLM, is that once again the GLM is estimating a higher proportion mature at younger but also older ages. And the A50 value, as you can see in region 2, between the 2 years, the A50 value has dropped almost by like 3 years. And so now if you see the GAM model, what you can see in particular for region 3, 2022, the year effect definitely smoothed out the model. So this is it's very important because it shows that extra years of data is helping kind of form the model over time. And as you can see, there definitely is there is a significant year effect, in particular in region 2 between 22 and 23. You can see that in 22 for region 2, there's definitely a more truncation of the ogive curve, showing lower proportion mature females, in particular between ages 10 and 15. But in 2023, those values definitely increased. But then in region 3, it shows a very similar trend. In particular, the A50 value is very close to the same between the years. So then to populate a coast-wide ogive using both 2022 and 2023 data, because there is a significant year effect in the model, and we don't know exactly what's causing that yet. We haven't been able to tease that out. To populate the coast-wide ogive, we decided to pool the data between years, which then generates the regional curves that you see on the right. And then, as we showed using the 2022 data back in June, to populate the coast-wide ogive, we used the most recent 2023 FIS space-time model abundance estimates to weight the regional curves. And that is what is populating to estimate the black line that you see on the right. And as you would expect, the line generally falls between regions 2 and 3, as those are the 2 regions with the highest abundance estimates from the space-time model. And this new output is giving us an A50 value of 10.3 years, which is over 1 year reduction compared to the current maturity estimate used in the assessment right now. And then this is the figure that Ian showed in a small little box in his presentation yesterday. But this is using the 2022 and 2023 data, comparing the GAM, the GLIM, and the current assessment models. So you can see how they're kind of differing in how they're estimating proportion mature for females for the coastwide stock. And as you can see, the GAM overall is a better estimator for proportion mature at younger ages, but also at older ages. Whereas the GLIM is tending to hit 95 to 100% maturity sooner than the GAM. But we know that not necessarily biologically to be true, because we are seeing that there are individuals 20, 25 years that are immature. Yeah, so this is just a good representation of how the model outputs differ. And then it look
Maybe just to remind them on the what we're using, the red curve that's being used in the assessment has been used for years, was just a logistic fit with a truncation below age 8.
That's right. Yeah, so to piggyback on that, so histologically, we have found that 7-year-olds do mature, whereas previously we thought anything before age 8 was immature. All fish were immature. But histologically, we are seeing that younger females are maturing. And so that's where the current assessment maturity estimate definitely disagrees. And also you can see that it's asymptoting at 100% a lot sooner than what we are seeing histologically. And then just moving on to the future. So this year we did collect histology samples again on the setline survey. We were able to collect samples out in the Bering Sea. So this will help improve our estimate in region 4. And then obviously, as you know, the sampling design was greatly reduced this year to only the central Gulf of Alaska, as well as Southeast Alaska, Reg Area 2C, and the northern part of 2B. So we're hoping to incorporate this information to hopefully be used in the full assessment for next year, 2025. So just in, I got all locked down. So in conclusion, so we are finding that the GAM is a better fitting model compared to the GLM when comparing over the 2 years of data that we have. The coastwide ogive, so we did pool across both years to generate the coastwide ogive, but we did see an observed reduction in the A50 value. So the curve has been shifted to the left between 2022 and 2023. And then in final conclusion, so we have sent out all the 2024 samples for histological processing, and we're hoping to get through those this fall and probably early winter. And the goal is to incorporate all 3 years worth of data for the full assessment in 2025. And then I guess, yeah, so some of the questions that Ian had brought up yesterday regarding kind of how we ultimately incorporate this into the assessment is kind of the real big question, because we have a long-term historical data set of our field observation data that's currently used. And we're not quite sure how we want to incorporate this new histological data through the historical time series. In particular, because we are, to create the coastwide ogive, we are weighting our regional curves, and that data can only be used back to 1993, because that's when the FIS index started. And so how we ultimately try to incorporate that historically back through time is really the ultimate question. And
Sorry, can you clarify the weighting is based on the average proportions by region for the whole time period, or just the most recent?
it's the most recent FIS index. Yeah. So for the 2022 and 2023 data, we weighted based on the 23 index, which is the most recent index that Ray has produced.
In the assessment, can you not use region-specific ogives?
No, because I don't have explicit spatial structure. I'm still
got the is.
Right, so for years where we have a FIS index, we can do the abundance weighting and I can create a composite that is weighted appropriately. But then before 93, it's unclear how to go. And it's not just noise. The stock distribution has shifted pretty dramatically over time. So it's definitely going to be important.
I think the most striking result is that age of 50%, age of maturity has shifted so much from one year to another. And it will be very interesting to see whether that keeps continuing when the new examples come, because we've seen through history that if fish start to mature younger and smaller, that is often some sort of early warning that something is happening.
Yeah, so I will note that in general, histology versus field classifications does tend to shift ogives to the left. And it's because histologically you're better to classify younger fish as mature, immature. Whereas if you were to see like a 6 or a 7 or an 8-year-old like on the FIS in the field, it's probably harder to classify it on the boat versus what I can see histologically. So that's just a general, but yes, we have seen, I mean, Clark and Hare, the paper that Ian is using for his maturity estimate currently in the assessment, they have looked at A50, L50 values from, I think it was the 60s, 70s, and 80s. And there is a shift there. So I think over time, there has been multiple shifts in where those values have fallen for Pacific halibut.
Could that be something to do with the reducing weight of age?
Well, what's interesting is that the maturity at length has changed dramatically as size at age has changed, but the
Yeah.
maturity at age has actually been quite stable
Mm.
over the 30-year period that Clark and Hare looked at, suggesting that either maturity is really dictated by how old the fish is. It doesn't matter when they get to a certain age, they just start maturing, or there's something more complicated going on there.
But no one knows when they mature how large their reproductive output is and how much that affects their body size.
That's what they're doing next year.
It's funny that I've met up with a working group that's a life history working group that's nationwide, and one of the chairs of that group, you just mentioned it. We like to call it the F word, and that's fecundity. And so that is the next step in all of this is looking at egg output and how that might differ as fish might be maturing earlier. Are they producing fewer eggs? You know, that's a question that needs to be answered. And we are collecting samples for fecundity, so that's the next progression in the cycle of our reproductive research.
What's the current assumption or pattern in the assessment?
Fecundity proportional to biomass. So the cubic function of length.
Cubic function.
Yeah, so no,
It's the
Length. So directly proportional
to weight, yeah.
so heavier individual is not proportional.
Yeah.
Correct.
so our current projection is that there's no potential like hyperallometry and like, you know, the BOF effect, basically big old fat fecund fish. We don't know if Pacific halibut follow that trend or not. It is found. The Northeast Fisheries Science Center has found, I think winter and summer flounder. They have found that the bigger they do produce, there is a hyperallometry. So we're interested to see whether or not Pacific halibut has that similar trend.
But I would be very cautious pooling years, rather pooling areas, but keep maybe years separate because there might be something interesting going on.
Yeah.
At least to present that pattern.
Yeah, obviously what we've presented today is, we can't say it's a trend. It's just 2 years, right? So we're hoping 24, 25 can really show us where the A50 and where the ogive curve is really falling over through time.
be interesting to see whether the regional differences in cardiogenic persist through time.
Yeah.
You know, whether the scaling just shifts for all
Yeah.
regions within your effect.
Yeah.
Or,
Yeah.
or whether they're really behaving independently.
Yeah, like we've seen in region 2, there was a pretty big shift, but region 3 was pretty consistent between the 2 years.
For 2 years.
Yeah. for 2
And there
years.
are some consistent differences in size and age between the regions. So region 2, which is showing generally a flatter curve over age, usually has higher size and age. So the fish in region 2 seem to put a lot of energy into somatic growth, which is consistent maybe with slightly lower transition to maturity, but
they could also be moving.
they move.
Bigger
Yeah.
individuals move farther.
Yeah.
So they could be in region 2 because they are bigger. Exactly. That's always.
Well, that's another.
That's why you might see changes in where your D50 plays out.
Yeah.
Depending on
how far
where they are and where you sample.
Yeah, movement is definitely, it could be big, an impactful factor in all of this.
Yeah, timing of sampling has not been evaluated much because the timing of sampling is fairly large. All the samples have been collected between end of May, beginning of June, and
End of September. Yeah. Beginning of
And,
September.
you know, there's a lot of biological processes related to maturation going on during that period of time. So it's very different. The proportion of mature females that you're going to find at the beginning of the sampling period versus the later part.
Maybe this is premature, but have you done any sensitivity analysis with hyperometry?
I have, but only in the context of whether it would improve the stock recruit relationship. So I ran a couple models where I added hyperallometry and looked at the root mean square error of the recruit Ds to see if it was actually, if there was any indication that doing that was a better estimate of spawning biomass, which would then make the curve tighter. And I didn't. But that's a pretty weak test. Somebody published a similar test though, so it wasn't totally.
People publish all sorts of stuff.
True, but we thought it was worth trying, even though it seemed like a fairly weak way to go about it.
Definitely.
I guess the crux decision at this point is we are thinking that 3 years of data will be enough to try and include this in some way in the next assessment. So that's as you saw from the sensitivity, it's potentially pretty important. What we're saying is all of a sudden a lot more 7, 8, 9, and 10-year-olds are mature than we previously thought, which makes things look pretty optimistic compared to the current assessment. So I think that's a huge. So the first hurdle is 3 years. Is 3 years enough data to actually go ahead and try and use this? And obviously, I'll look at this in several different ways in the assessment, try to understand the best way to do it. And then once we get over that hurdle, the question is some of these other things, like if we use the abundance weighting on the curves, do we go ahead and use data from 3 years, but use the abundance estimate from a 30-year time series, and then do some sort of average for the historical period? And there's a lot of ways we can put that together. We can do some sensitivity, but we won't be able to do every sensitivity between that and everything else we're evaluating in the model. So we're going to have to make some decisions about how to proceed on that front.
Two things that are really on our plate to examine is obviously we mentioned the work on fecundity that's starting in 2025. The actual work, but in fact in this fall we're actually going and collecting samples for fecundity. So we have a dedicated effort to bring in samples to construct an autodiametric curve. So we're going to use the autodiametric method to calculate fecundity. And that's based on the relationship between oocyte density, number of oocytes per gram of ovary, in relation to oocyte diameter. And you need all the ranges of oocyte diameters possible. So I think that with the collection of samples this year in 2024, we're going to have enough range of oocyte diameters to construct that curve that then will be implemented starting in 2025. So in 2025, we're going to have the first results on. First, we need to construct that calibration curve method, and then we're going to start measuring fecundity in the samples that we have already. So that was one. The other part that is we still need to address is the the error involved in the macroscopic staging compared to the histological method. So basically comparing the macro and the micro classification criteria and which error is and in which classification category the errors are located. We have a broad idea of where the error could be, but that needs to be analyzed. So we have, fortunately for all the histological samples that we have also the macroscopic staging in the field. So we'll be able to compare that, but that's coming up next.
Can you tell from the histology whether an individual is first maturing versus mature and just maturing that year?
There's multiple indicators of you being able to do that. One is post-ovulatory follicles, so POFs, which a lot of post-spawners, they'll have residual POFs. So in a particular, a lot of times those eventually get reabsorbed over time. But if you, like for Pacific halibut, if they spawned in the winter, January, February, if we're collecting samples in the summertime, a lot of times you can see residual POS from the previous spawning. And so you know that has been a past spawner. Another thing that we're also going to be looking at is we did a little pilot collection on the FIS this year of taking some gonad wall collections, because there has been shown in fish species that the gonad wall thickness changes over time as the fish matures. And so we're looking to see whether or not that holds true for Pacific halibut. And that could be another indicator of previous spawner and an already mature individual.
Because it seems like what we see as an ogive is really two processes, right? You have individuals that are maturing the first time, and you have other individuals who are maturing this year. And we're just calling it proportion mature. Wonder, just spitballing, if there's any value in thinking about it as more of a two-stage process.
Yeah, I mean, it's something we could look at. I'm not sure if I've ever known anybody that's done that. I think.
For the year at first maturity is analyzed.
Yeah. I'm pretty sure all the current histological data that's being produced from anyone it's just under the assumption that the fish is mature, whether it's a first time mature or an already mature individual. But it's definitely something that we could look at. Yeah.
Because to me, I mean, these things seem very sensitive to year of first maturity. So even some of the gams, I don't think completely resolve those young fish who are pretty consequential for the spawning biomass calculation.
Yeah.
And then it's the thing, the maturity associated mortality increase. So those old ones that are not mature might dominate the delta, because some of those ones who have matured like more than 10 years ago, they might have died. So the data can get biased in the end. And that might explain why the GAM has this huge uncertainty downwards at the very old ages. Like the
Literally
survival cost.
survivorship bias.
Yeah, survival cost.
It's not really biased though with regard to trying to determine what the reproductive output of the stock is in a particular year. It's just biased relative to the starting distribution of the code.
I don't understand the underlying mechanism.
So Pandora's box is open. Here we go.
You got some room anyway. Fill it up. Okay.
All right, we'll pass it on to Claude. The next research area that he's going to provide some updates on is on the discard mortality rate in the entire recreational fishery and response to a particular comment by the SRB. Go ahead.
Thank you.
There we go.
There we go. So this is just a brief update in response to request at SRB025 to look at the recovery status of fish and how some of the parameters we've been looking at to characterize the discards, how they played out. And so I've presented the first three columns before, but essentially by row, these are stress indicators that we looked at in blood plasma. The first column is glucose, looking at how glucose changed against fight time. The viability category. So the viability category is what the animal is characterized at release by the observer methodology, whether it's in excellent condition, moderate, poor, or typically moderate, poor, or dead. In this case, there was no dead fish released, and we pooled the moderate and poor because there was so few observations. We looked at the effective injury type on the blood parameter, and then the last column is the new one, and that's looking at recoveries of fish. We've had quite a few recoveries of fish. Speaking to tagging recoveries, this study is having a higher rate of tag recoveries than we've seen in most of our tagging studies. Right now, we're at we were at the time of this presentation, we were at about 12.5%. We've since had another 3 recoveries in the 30 days since this was produced. So we're still getting recoveries out of the fishery. But that last column there shows fish that what their glucose levels were when they were released. So those that are still at large are in the left-hand part, and those that have been recovered by the fishery or if a fishery are on the right. And there's no differences for glucose. Same thing for lactate and cortisol. So those populations are not showing any significant predictive value to us. Similarly for fish length, Fulton's, K, and fat percentages, we did the same thing for at large and recaptured. We did get a significant size effect on the recapture, and that is likely explained just by the fishing regulations. Now, granted, they're allowed to bring these fish in no matter what, but that doesn't seem to be well understood by the fishers. We get lots of confusion on that, and probably could use some more outreach on that particular topic.
Does it say anything on the tag about that?
It doesn't say it on the tag. It says it in the regulations, and we talk about it at most meetings that we interact with fishers. But we've had, there was a comment earlier that there was a couple of recoveries that, I forget the gentleman's name, that you mentioned in the side there panel there that he's, occurred. A couple of those were captured, the tag was removed and the fish was released again. Or in one case, the tag was recorded and the fish was released again, which they're not being asked to release them. But sometimes those fish are not inside the regulations and they're paranoid about showing up at the dock with it. Or if they remove the tag and then enforcement can't see that it had a tag, why is this fish in your possession? There's a bunch of challenges there that I think create barriers. But at any rate, we did see a size effect on the recaptured fish, but we didn't see any predictive value to Fulton's fat percentage or things like that on their recovery. But we are certainly seeing a high percentage of recoveries on these fish. We're getting some valuable information on growth by fish size and things like that as well. So, and so that's it for updates on that. The rest of that work is being written up right now for publication on discard mortalities, and the satellite tag component of that showed a 1.35% mortality rate on excellent condition fish. So they have very high survivorship. And we're continuing to recapture tags that launch their satellite tag. They leave behind a stalk and an anchor in the fish. We're continuing to recapture those as well. So almost like a mark, recapture, release, mark again when you get, it's like multiple recapture type event.
Right, you get 2 locations.
Yeah.
All there any questions regarding to this work before we move on to the next?
So then just a quick update on fishing technology. We don't have a lot to report here. I'll report on the catch protection work. And we've also been participating in some work being done by the Pacific States Marine Fish Commission that have been looking at artificial light to reduce bycatch and circle hook modifications. I won't be reporting on that, although that work has been continuing. And if there are questions on what we're doing with that, I can speak to it. The catch protection study, we have grant money to go out and continue further testing on the shuttle device. This is the device that slides down the ground line, fish enter one end, they're removed from it, and they ride up inside the protective device to prevent whales from eating them. We were unable to secure a vessel to actually do the work this year. We put it out for bid earlier in the year and couldn't find vessels with available time to do the work. They were either fully tasked with our work or more typically with sablefish. There's so much sablefish quota that the boats were like, okay, once we start going after a sablefish, we have to catch 80% of it because the price is tanked, that sort of thing. And so we've secured a extension to that study for the grant. So the money's still in place. We've developed the camera equipment that's going to be on the equipment. And we're hoping to get the project again on the grounds in the spring or summer of 2025, hoping to pair it with some of the proposed work in area 4A, 4B, where there's quite a bit of whale depredation. We could hopefully make a more attractive body of work by having two projects available, both this and this project, to give more meaningful work for a vessel to travel out there and do the study. So that's kind of the status of that. It's on hold and we'll push forward to next year. Any questions on that?
Is bottom contact an issue
With the device?
with this fishery? I know in BC, there's some concern. I think the Marine Stewardship Council looks at your bottom contact, sensitive habitats, and things like that. Have you guys done any of that?
We're excluded from some of the sponge reef protection areas in BC. Survey. I don't know in terms of the limitations on the fishery itself. I don't know exactly the status.
So the fishery is excluded from those sponge reef areas, similar to what we are on survey. We do have pre-closure survey data in those areas, and so we argued that it might be valuable to see the effect of the closure by continuing them as we stay in a very tight footprint. But they declined our opportunity to go in there. So, but there is data before the closure, so there may be some value from our data sets going forward on that. And certainly, I've heard of questions about long line gear on the bottom in Alaska, but to my knowledge, there's no ongoing studies or specific restrictions. There's some concern over that in area 4, where there's quite a few coral communities, ecosystems. So
It's just so mired in protected area stuff, and there's all kinds of arguments that fisheries are damaging stuff outside of the sponge reef. So there's a lot of concern there. When the sablefish fishery went through Marine Stewardship Council certification, they slapped the condition that they didn't know the impact of the fishery on habitat. We're like, well, it's mud. And they didn't buy that. Ended up starting a research program on the whole thing. It's produced a lot of interesting results, like some of the current maps of coral and sponge distribution are wrong because they're taken from opportunistic trawl samples. So if you go down the bottom cameras and look around, you get a different picture. And so that impacts the footprint. So, I don't know what, if it's not really an issue in Alaska, but to be, it's
Sean, was your question related to whether this catch protection device actually made an
no, I'm
impact
just
on the
thinking
bottom?
like in terms of fishing technology, in terms of a technology kind of research program, if There's a lot of tech involved in trying to do that.
Well, we participated also with Pacific States Marine Fisheries Commission. I mean, Ian and Claude will know about the semi-demersal.
Right. Yeah. So one of the things that Pacific States Marine Fishing Commission has been focused on is reduction of yellow eye bycatch in various longline fisheries, including the Pacific halibut fishery. We've done a bunch of work on what we call hook appendages. So these are traditional circle hooks with a welded appendage, a wire appendage sticking off to one side or the other. And it's been found that a 45-degree angle appendage effectively reduces yellow eye catch while maintaining Pacific halibut catch. And it's essentially thought to be a physics or biomechanics issue where the animal comes up and goes to bite it. If your mouth is oriented one way or the other, it either bumps it out of the appendage bumps it out of the way, makes it less catchable than the other. The other one that they've been looking at is a concept of semi-pelagic gear. This is a practice that's been done particularly by the sablefish guys, where they will they'll essentially float the gear several meters off the bottom, and predominantly to avoid sand fleas and hagfish from damaging the catch. And so the thought was maybe we can catch halibut up in the water column and less rockfish in the water column. And is that something that we can test out? So the testing for that happened this past summer, and it clearly showed that there was no benefit from doing that. We were catching, if anything, we were catching slightly more yellow eye on the floated gear than on the spotted gear, but it was not significant in either test. So it's not showing any differences. No benefit to doing that. The benefit probably comes from reduced market damage to those fish. So that was something that, foreseeably, if you were in a sensitive area, you could float your gear. And essentially what they're doing is they're putting styrofoam floats on the ground line that helps lift it up off the bottom, and they're anchoring it to the substrate in certain spots, but you'd have less of a footprint touching the full substrate. Even when you catch a fish, it's not going to be whipping the ground line around, the canyon around, and damaging other stuff in the area. So it is something that can work, but overall the catch rates were quite a bit lower than just leaving it, demersal right on the ground. So I think that would need some further studies if that was a viable technique. It's something that's used by a handful of fishermen, but it was not effective for that purpose. But you could foresee it being a technique used in sensitive areas to reduce your footprint.
So the last slide is just to note the two research projects that the Secretariat is conducting. One is the bycatch reduction engineering program that Claude just mentioned. This project has just, as you heard, has received an extension. So this project will last until April 2026. And starting January 1st, 2025, we're going to start with a new Alaska Sea Grant project that is based on the development of a non-lethal genetic-based method for age in Pacific halibut. That's to develop an epigenetic, what's called an epigenetic clock. That's based on DNA methylation patterns on the genome that are associated with age. So that's an alternative method that we're going to test to see if its feasibility and eventual complementation to other existing methods for aging. So that's starting in January. And that's all we have.
So the 60,000 for that, is that how much progress do you make for $60,000?
Well, this
This
project is in collaboration with Alaska Pacific University. This project, Alaska Sea Grant has a cap of under $100,000. So these are projects that are not extremely well funded, but the counterpart is that with the project, there's stipend and tuition for a master's student. So we've already recruited a master's student that's going to be working exclusively on this project as her master's thesis through Alaska Pacific University. So the $60,000 really is for consumables for us and a small component of our salaries, Andy's and myself. So it's sufficient for the purpose of just developing the technique, not for running, the samples. The project, I think I shared it in the last June meeting, the proposal for this project, the PDF. I can resend it again. Proposal was submitted, in fact, in 2023, and Alaska Sea Grant has been extremely slow in awarding the award. So finally, we've got it now, so that IPHC has a sub award from APU to conduct this work.
I guess you have having the genome matters for that.
Yeah, well, it's essential to have the genome and the reference genome for the study. So. That's all for us.
Okay. Okay, well, that didn't take 3 hours. Some good progress here, or so we could have some open discussion for. Why don't we have like an open discussion for 15 minutes? Then we'll put some coffee breaks.
Should be then drop.
Even go better. Drafting.
Yeah.
We have a lot of new stuff.
It's good to know. Have
You want to do some more thing?
you ever written some notes?
Yeah, we could do that. We could, and then get back on tracks for lunch or after lunch. Okay, so one of these, just 15 minutes or however long, doesn't matter. On that, we'll get the draft up and have a drafting session until lunch. And then after lunch, we'll get into the monitoring.
So back in 15?
No,
For us.
we have an open discussion right now for 15 minutes. My big question is, so for the ecology biology program, like what's the big thing coming? It's been there's been a lot of progress on things, which is going to leave a big.
Yeah, I
What's the
think
next big thing?
for 2025, what's coming is the completion of the third year of maturity assessment. So incorporating the third year, the 2024 samples into the 2022 and 2023 data. So we're having a 3-year and then providing that data for the full stock assessment. Number 2, in terms of importance, I think it's the fecundity estimations. So the development of a method for quantifying fecundity and then starting to look at fecundity in different biological regions from samples that we already have. And we're planning on a collection of samples, additional samples for fecundity in 2025. We want to make sure that we also capture, the larger components. So we're going to be targeting particularly large females to determine if there's this hyperallometric relationship between weight and oocyte production, egg production. Number 3 in 2025 is the delineation of immediate plans, possibly linked to a research proposal on the topic of size at age. On the topic of size at age. This is a topic that was touched upon in an NPRB, so North Pacific Research Board funded project that finished in 2021. And we've have recently a series of discussions to see what components of that study could be continued and capitalized. So we're looking into delineating some ideas for a proposal, for a research proposal that is intended to better understand spatial differences in size and age. And that's based on some unpublished results that suggest the presence of different hotspots within the Gulf of Alaska and cold spots in terms of size at age. There's areas where on size at age, looking over, and Ian can speak to that better than me. He's a co-author of that study. And that provide different views into size at age within the same general. So those ideas have not been consolidated yet, but we're having a discussion. We had a discussion on Monday that included Dana on that topic, and we're putting those ideas together to see how we can advance our better understanding of spatial differences in size and age, particularly in the Gulf, and whether we can actually incorporate some of the gene expression data and some genomic data in that analysis to understand whether there's a genetic basis for differences in size and age, or whether there's an environmental component driving different levels of productivity in different areas. So those ideas are still not very clear. We need to continue developing those. So that would be the third project really that we want to accomplish in 2025. The fourth one would be the completion also of a study that was mentioned by Claude, and is the completion of a catch protection study to see whether there's an advantage of using catch protection devices to minimize whale depredation. That has direct impacts on the fishery and also on our fish. So I think those four areas, and in addition to completing the work that is ongoing, one of which probably one of the most important ones for publication is Andy's work on population genomics. That the target is to complete that study, publish it within 2025, and then determine, once that's published, determine what's the next step in ability to use genomics to understand population dynamics. There were some interesting aspects from Andy's work that could be followed on. They need a little bit of work delineating which ones are those, particularly the temporal differences across years. We're not seeing much spatial structure, but there were some temporal differences that were at a few SRB meetings ago were highlighted by Kim Scribner as potentially interesting. We may want to look at those. I think we've got quite a few topics on 2025 to keep us busy. I'm not sure about this.
Does the size at age study include? But I was going to ask because I recall those. There's a bunch of work going on what was driving growth.
Yeah,
growth studies and things.
Right.
I mean, changes in size at age are, aside from movement being a huge uncertainty, changes in size at age is probably the other one.
Yeah. So I think it was it's timely to revisit that topic and delineate research strategy. Actually.
I mean, I ask because I guess you're doing research strategy. So that would be a part.
So related to the DNA methylation pages, I understand there are some DNA methylation markers, the term right, that can be used as indicators of cumulative lifetime stress. So, that they don't necessarily accumulate at a set rate, but accumulate faster under more stressful conditions. So, is that something that's developed enough that it could be looked at here, or is that still?
I followed that literature a little bit, and yeah, I don't see that aspect being developed yet. It's potentially
too
interesting.
experimental to be applied at this point.
Yeah, there's a few things that are potentially interesting in addition to, setting that relationship between methylation signals and age to establish that chronology, that genetic clock. One is better understanding of senescence and specific element linked to a number of biological projects. Anna just mentioned the large females, maybe not mature, and mortality associated with those. Maybe there's some methylation signals that could shed some light as to what is the potential survivability of those fish that reach a certain age. Maybe there's a difference rate of survivability or mortality associated with males versus females according to an age senescence. So there's a few things that you could learn. I mean, the advantage of that method is that actually you're looking at a biological process that's driving things. So maybe I'll understand a little more in addition to age.
Yeah, I asked that partly because I think that the value of that technique for aging is not huge for halibut because you've got really accurate aging methods already.
Right.
So having another aging method doesn't seem like in itself can be useful. I mean, yes, you could do it without killing the fish, but this is not an endangered species. You've got plenty of dead fish to sample.
Right, but for instance, all the tagging work that could be incorporated in all the tagging work. But actually,
You get
ages about
You get an
the
age of
basis. So
Okay.
that's there's definitely an advantage of having that method. Yeah. So yeah, and you see in the literature more species for which those genetic clocks have been developed. That's definitely on the horizon as well.
I noticed there's something in the chat there. I think we read that.
Just related to tax.
Was that Kurt's comment?
He had a second comment about a charter operation that was illegally tagging halibut. And then eventually enforcement caught up to him because of tagged fish by the IPHC is a free fish. You're not allowed to just go tag your own fish.
For the clarification, under IPHC regulations, we can permit individual projects under which fish can be tagged, but this requires applying for that permit and specific conditions for allowing external tagging. And then these fish are not subject to the same rules. These tags, the fish tagged by not the IPHC, then they're subject to the same rules as whatever fishery.
Yeah, going back to the topic of size and age, one of the things that the Secretariat had worked on for quite a while is determining whether you could actually use autolytic growth increments to recreate individual growth trajectories. And that's something that we don't have an answer for. We've done quite a bit of work in measuring growth increments in 4 cohorts that experience different size at age. Starting from I think one of the first ones was the 1987 cohort, up until much recent ones, comparing males versus females as well. Because, there's a more dimorphic growth. census. So that is something that we may also want to revisit and finalize whether actually use that method to track individual growth trajectories or not. Yeah, there was a question whether that was feasible for Pacific Halibut. We were discussing that yesterday. That there was a relatively recent paper on Atlantic halibut that they seem to use those growth increments.
They said like reasonable.
Reasonable.
Whatever, you know.
We just need to, we have the data. We just need to finalize the analysis and then write a publication. Whether that's a useful way of backtracking growth individually for Pacific halibut or not.
Determine whether it's useful or not. I mean, you can go ahead and do it, but
Well, to see if it matches the size of age that those cohorts experience, and also whether that can basically track the growth differences between males and females. And there was a question whether we didn't complete the analysis, and that's something that we might want to revisit and then put a lid on it, and then if that's a useful technique, then we'll go ahead and use it. And if not, report it as such.
I guess I'm still not totally clear on that because I'm interested in this method as well. I mean, what you want to know is whether the growth increments are proportional to the growth in that year. Right. But how do you verify? How do you verify that?
Well, a lot of it is based on a linear regression relationship between otolith size and fish size. And that's how they estimate growth rates, back calculation of growth rates. So
The assumption is that if the otolith size is linearly related to.
yeah, now I did run into a former lab mate of mine who now runs a lab at UC Davis, and they do a lot of otolith work and otolith geochemistry work. And he's going to actually forward me some papers. So not every fish species follows that otolith or that otolith trajectory. But people have there's still a lot of studies out there that have shown people being able to use the otolith to back calculate growth rates from fish. It's
Even if it doesn't.
even if it doesn't. Yeah. He said if you can still build a certain type of relationship, you might be able to model it out in certain ways. He says there's assumptions that have to be made. But there's a lot of work out there that's doing it on a bunch of fish species.
Oh yeah, it's been around for a while.
Yeah. So I mean, and then I was telling him, we have such a huge historical database of archived otoliths here that go back decades. And so if there's any way that we could figure out a way to potentially use those to back calculate growth rates through time, I mean, we're talking 100 years worth of otolith information that we have here. And it could really show us different signs of how the size and age of the fish has gone up, and then now it's gone way back down. And I think it's a gold mine for the IPHC here if there's a way that we can figure out a way to use it.
Yeah, I agree. Just trying to understand the confirmatory analysis that you're going to do.
Yeah, I mean, because I've done it with like early settlement young a year fish, like in California, and a lot of those, it's like, I mean, you're talking like R-squared values of like 90.95 or higher. They really show up for otolith size and fish size. But then once the fish, I mean, we were just talking about this last night, actually, about I think eventually there's a point where because Pacific halibut gets so big, the lobe is so big, the otolith eventually it can't just keep up with growth, right? So I was talking to my colleague about this, and he was saying that a lot of times in the bigger fish species, the otolith length doesn't get bigger, but like the calcified carbonate starts to just overlay on top of the otolith rather than expanding like lengthwise. And so he was just interested in the things that we could do here because of what we have, the data set that we have here with the archive.
So did you look at the body size related to otolith size?
Well, that's something that we have to, so there was a study done, I think it was like 7-8 years ago, maybe. I think they started here we need to go back through the data and really look
You
at it.
don't have those data just in the spreadsheet.
We do,
Yeah.
we do have the data. But none of us, I don't think any of us here in the office have, the person that was doing it is no longer works at the IPHC. So none of us, I think, have actually like gone back and looked at the actual, like what it would look like, what an R squared value would look like for a regression of otolith size to fish size. That's something that we want to revisit, to look and see whether or not there is, what the fitting relationship is and how we could potentially use that to back calculate growth rates on halibut.
There's a lot of historical work on this topic for halibut. So in the 1960s, they determined that there was a tight relationship between otolith size and fish size. So they stopped collecting lengths, just collected otoliths, and then we were inferring the length from the otolith size.
Wow, that's risky.
And then in the early 90s, Bill Clark reevaluated the relationship and said, guess what? When size at age changed, this relationship changed. So this isn't working anymore. So we started collecting lengths again on those fish.
Yeah.
So
that's the downside. I mean, thinking on
Right.
a length.
Seriously. Well, we were sampling derby fisheries that were sometimes only 24 hours long. And so sampling a lot of fish was deemed to be more important than more information from a few fish, but it turned out to be not the right choice. The trouble really, I think, is that both the assumption that otolith size is proportional to fish size and the assumption that otolith weight has been published several times as a good proxy for fish age. But one of those can't be true if size at age is changing, right? And yet both of those fields of study sort of assume that it's true, that otoliths get heavier as fish get older and that otoliths get bigger as fish get larger.
Well, those patterns should generally be the case, but
Right.
that doesn't.
But
mean
when we
that
have
predictive
huge
power.
size at age changes like halibut, one or both of those have got to break down. So I think we just haven't figured out how to deal with that. And even if we do a short study on fish size otolith increment relationship, it doesn't mean it's going to hold up as a global relationship. Right. As we've learned the hard way.
Good. I suppose you could at least look at the big tagging database as well. So if you had something that was at large, so you measured it at tagging, and then it was at large for a year.
Yeah,
Some
we
idea,
have a
like-
few issues with our historical tagging. The most disappointing one is that for quite a few years, they were concerned that when we got fish, because occasionally there's measurement error, right? So occasionally you'd measure a fish at time of release, you'd measure it at time of recovery, and it could be smaller. But that was deemed not realistic. So they corrected one or the other of the lengths, which mean, and it not in a systematic way. So it means that it's very difficult, if not impossible, for us to estimate the measurement error and the true increment at the same time. The way the data was. They didn't store the original data.
That's very commonly.
It is.
Measurement error.
Yeah.
Just-
Right.
I've looked at a couple of tagging data sets for striped marlin in particular, hoping to use that. And unless you've got scientists measuring on both ends, it's really hard. Constituent-based tagging programs, you never get good ones to.
We should have some more recent internet data that won't be subject to that. We would discontinue that practice, but unfortunately, the bulk of our historical tagging is subject to that issue. And unfortunately, for most of our historical tagging, we know fish size, but we don't know fish age because it was recovered in a situation where we didn't get the otolith back. And we don't know the sex because we didn't have a genetic sex technique. And most of the recoveries come from the commercial fishery where they were dressed. And so because of the dimorphic growth, patterns that we see in terms of like migration by size, we can't unconfound from differences in males and females. So we are tantalizingly close to these incredibly rich data sets, but missing some key pieces in them.
The otolith growth increment analysis, and maybe this is where you're going, that's most powerful when you tie it to environmental correlates. Look at developing predictive relationships between temperature, most commonly, or some other aspect of the environment.
Especially if you're doing this study in spatial variation, size at age. But it sounds like there's pockets of tools that are now coming available that you just have to let the old stuff go and start building a new data set, the ideal one for the questions you're asking. now that you can, if the epigenetic tag aging works, then now you've got that to work with.
And we get sex from fin clips. So as long as we get
Sex
fin
pin
clip
clips,
back,
so there's a lot of those things have been resolved. So I'm not sure we have a whole lot to add to the report this time. So Anna suggested maybe that we just move on. Just throw the drafting. So Ray thought he was gonna have a couple hours off. Quick break. Yep. Let's take, yeah, let's come back. A long time, but we all can occupy ourselves for 20 minutes. Okay, so back at 11.
Okay, well, welcome back, everybody. We are moving on to agenda item 4.2. Under 4.2.1, we don't have any particular items, the fishery dependent data. But under 4.2.2, the fishery independent data, we're going to hear from Dr. Webster on paper 09. So you can have that presentation brought up, please. And then over to you. For sure, or you, yeah, go for it. No, still for that. Okay, let's see.
You want to have that blue arrow?
Oh.
I saw it in your presentation.
Click.
Oh, it's that,
Yeah, I
Finger.
think that finger happened. May not. have
Maybe I
moved,
can move
just move
so it comes back as soon as you, okay,
Oh,
whatever. Okay. Hi, for those online, I'm Ray Webster. I'm talking about this again. The main reason for bringing this back is from, we presented most of this material in June, was explained by the observers yesterday, that the commissioners really want the SRB's feedback on scientific aspects of the survey design that are under consideration. And so we're bringing this back to give you an opportunity to make further comment for the benefit of commissioners and for ourselves. So I'll start by reviewing, doing the usual review, but I'll try to move quickly because as I said, most of this was presented back in June. And you heard about this yesterday from Ian and Ellen as well, right? About the FIS. So we use the FIS for estimating stock trends, distribution, getting data for the assessment. We've had an annual FIS since 1993, expanded over the early years, and again over more recent years. So we got this full design of 1,890 stations indicated in orange here. We supplement this data in the Bering Sea with data from the troll surveys, typical full condensed troll survey design. These are stations we use for our data purposes, shown in red there. But we can't sample this full FIS grid each year. Logistically challenging, cost prohibitive. So we've been prioritizing sampling effort based on scientific needs and long-term revenue neutrality. This lays out the objectives. Primary objective is scientific, dictating the station distribution, station counts, skates per station. Secondary is long-term revenue neutrality, cost, logistics and cost, operational feasibility, and cost revenue neutrality. And the second objective is the one that's been placing some serious constraints on our ability to fish the FIS designs that we wanted over recent years. We have a design review process, and we are currently, as part of the process, we presented some preliminary designs with costs for 2025 to the commissioners earlier this month, and we're presenting things to you again now. At the moment, this data have been finalized because we had a shorter survey, so things were finalized early. And so I'm at the moment running the space-time models on the current year's, including the current year's data. We're still waiting control survey data from the Bering Sea. So it doesn't mean we're going to have everything finished early, unfortunately, but most we'll have a good sense of where things went, how things changed. So the recent history, in 2020, a rationalized FIS design was approved with random sampling in the core areas, 2B, 2C, 3A, and 3B, and sampling of blocks of stations in order to have more efficient sampling in areas where it was more costly to maintain precise estimates with low bias. Unfortunately, the COVID-19 ended up reducing the FIS that year to the core areas only, 2B, 2C, 3A, and part of 3B. The subsequent 2 years, the proposed design was largely implemented. We reached the fish what we wanted, except Western 4B was not sampled in 2022 due to lack of viable bids. And that is an area, as Ian mentioned yesterday, that doesn't have a lot of commercial sampling, so we don't have much information at all in recent years from Western 4B. It was last sampled by us in 2019. And as yet, there are no plans to sample it in 2025. 2023 to 24, reduced designs implemented to lower costs. We had little sampling outside the core areas in 2023, with no sampling in 4A, 4B, or 4CDE. Further reductions 3A and 3B this year, but some sampling was added in 4CDE. So this was the implemented 2023 design. You can see extensive areas with no sampling at all. We had planned to sample here, but got no bids in 4A, 4B. Only a small amount of sampling in the far north of 2A and gaps in 3A as well.
Ray, they can't see your cursor unless
They
you
can't
pick
see
up
this
that blue
cursor?
one online.
Okay, that's why I started describing the areas as well as wiggling around the mouse to drag this thing around. Okay. The adopted 2024 design was scaled back a lot, and I believe you've seen this already. We sampled essentially the revenue positive parts of 2B, 2C, at least we thought that's what they were at the time, and 3 charter regions in 3A, 3B. But we also, the commissioner has directed us to add the 4CDH, southern 4CDH, and island stations, which gave us some information in that region, which we haven't had for a couple of years. So in recent years, the FIS has fished around stations in the core, as I mentioned. Well, that was the goal anyway. This method was chosen in 2019, a proposal to fish charter regions as blocks of stations, similar to what we do outside of the core. And then the commissioner has since directed us to evaluate potential block designs, and that is the preferred approach for us going forward to improve efficiency of the FIS, hopefully get some lower bits relative to designs based on randomized stations. So as I mentioned, we've got a base block design, which is designed to have high spatial coverage over a short period of time. Not all parts of each area are fished every year, but the parts that are rotated over a 2 to 3 year period in the core. So the core is covered completely within 2 to 3 years. Elsewhere, we're prioritizing coverage to minimize bias potential and maintain precision. But this design, which you'll see maps of in a minute, is designed to meet our scientific needs. But not all regions that are currently revenue positive are sampled each year in this design. So if we're rotating through the blocks, sometimes we may be catching the least profitable ones or the ones that make greater losses. So there's potential to increase revenue if necessary by increasing sampling, as we've done in the past recent past by increasing the number of stations we fished within the randomized design in order to increase revenue. So these are the base block designs for 2025 to 2029, should they be implemented. And you see that pretty much the entire coast is covered over that that 5-year period. There are some exceptions like Northern California, which is not high priority because of low density, the inside area 2B, but otherwise, over a relatively short period, we have very high coverage, leading to low potential for bias and maintaining precise estimates. But in the last couple of years, the base block design has been projected to result in substantial operating losses for the FIS and would require supplementary funding to implement. So we devised a couple of other designs, which again you've seen in June, but they were discussed yesterday. The core block design maintains the same rotating block coverage in the core areas, but removes sampling outside of the core areas. Again, not all revenue positive areas are sampled each year, so there's potential for increasing revenue within that design. The reduced core only samples this charter regions that were planned for 2024, so it's meant to mimic a highly scaled back design like the one we implemented this year. It's meant to be representative of similar low coverage designs, noting that the more profitable charter regions will vary over time, although if we're not sampling them, I don't know which ones they are. So that's the core block design 2025, same as the base block, but with nothing outside of 2B, 2C, 3A, and 3B. So this would result in good spatial coverage in the core over a short period of time, low potential for bias, precise estimates within the core, decreasing precision and increasing potential for bias elsewhere. Would feed into regional and coastwide estimates as well. The reduced core design is simply this, fished over 5 years. Again, that's a representative design. It would be unlikely that that would be what would be implemented on the basis. So in summary, base block design projected CVs of 25% or less for all regulatory areas, 15% or less for the core. All biological region CVs projected below 10% except for B, low coastwide CV. So precise design, but more importantly, as Ian mentioned yesterday, we're trying to move a bit away from the focus on CVs. They became a focus, they became a sort of a target for us to examine for criterion for evaluating designs back when we had much more even coverage across the coast. And so if we had low CVs, there was potential for increasing station density or doing something to improve that in a regulatory area. Where now we have large gaps in coverage, we're more concerned about the bias induced by those gaps in coverage. You can still have precise estimates while bias increases. But with this design, it should mitigate the risk of bias while keeping estimates precise. Core block, again, we're doing well on the core, hence the name, but rapidly increasing precision, decreasing precision outside of the core. So not projected to maintain precise estimates outside of the core, but also the risk of bias increases over time. The bias is also expected in the age composition information, as well as stock change and stock distribution. The reduced core, this time we're sampling less frequently within we're constrained to where we're sampling within the core. So the CV start increasing in core areas, except for 2B and 2C, where coverage still remains really high. We now have increasing bias concerns within the core of the stock as well, particularly region 3.
What's the coastwide CV?
I'm sorry, what?
What is the coastwide CV end up being?
In this one? Yeah, I've got it tabulated in the extra slides. Yes, it's like the first extra slide. It's still relatively low, but again, our concern is the bias at 8% up to 3 years, but it's increasing. Yeah, if you were to look at just that, you'd think, well, this is still pretty good, but it's not. So in terms of revenue and cost, the designs were optimized for numbers of skates, 4, 6, or 8 skates used, depending on projected catch rates and base costs. So one of these is going to be optimal in terms of revenue or loss. We made assumptions about price and catch rates declining 5%. I don't know why I kept this one in. We are planning to use just chum salmon on the 2025 survey. I think salmon didn't provide us as big an advantage of, in terms of price as we expected, and the catch rates turned out to be lower on average with pink salmon. They tend to stick on this well for most people. Folks in 2C seem to handle it well, but elsewhere not so much. The CCAD is our oceanographic monitoring tool. And so whether we use these or not, there's an additional cost associated with them. With the CCAD, without the CCAD, we're still looking at a deficit of over $2 million for base block design and even the reduced core. So when those numbers are higher than what you saw in June due to revised costs, and these have been revised since then as well. We're in the process of being revised. Okay. I'm going to skip this one. It's just basically a summary of why we would be concerned about using the core block and the reduced core designs due to this information. Impact on the stock assessment. The slide also appeared in June and this takes a long time to talk about. So more interesting perhaps is where are we at in 2025? And this is, for now, I call this the starting design. There are probably better names for it, but this is perhaps the most fiscally viable design that ensures coverage in all IPHC biological regions. So we have one charter region in each of 2B and 2C. These are projected to reduce losses relative to no-FIS. And if we run no-FIS, we still have fixed headquarter costs, staff employed here working on the FIS. So by doing nothing, we lose more money than by going out and fishing the areas that make us some money. And those are those two charter regions here. The Charlotte charter region in north of 2B and the A charter region in 2C. In addition, commissioners have secured supplementary funding for 2A and parts of 4, the priority parts of 2A and parts of 4A, 4B around the border. So we talked yesterday about concerns with those areas, local concerns with those areas, but perhaps in particular for 2A, it's a very small piece of the puzzle and doesn't have a great impact whether we have data there or not on our overall understanding of the stock. And those local concerns have influenced commissioner decisions, of course, and they hear from stakeholders, and so the priorities might be a little different from the broader scientific priorities that we have in the assessment. And so they've prioritized, they've made supplementary funding available so that we can get a survey in 2A in the highest density parts of 2A and also the highest density parts of 4A and 4B here. So there'd be 60 stations at each part, each forming 2 additional charter regions. Commissioners are also going to be presented with a series of charter regions made costs in case additional funding becomes available. And I skipped this part, but these are the 2 charter regions in 3A, 3B that we expect to make the least loss. And so I don't believe they will know more about this, and I'm not sure what can be said, but I assume we're hopeful that funding or some mechanism for funding those becomes clear in coming. Nevertheless, this design, while maintaining coverage in all IPHC biological regions, and if you include the Menstrual survey, we have coverage in all IPHC regulatory areas, has substantial gaps. We have 366 stations out of 1,890 stations, where previously we typically fished between 1,000 and maybe 1,300 stations a year. And in some cases had a randomized design which provides greater spatial coverage even with fewer stations. So this isn't optimal. Looking at other options, one issue with this design is that these 2 areas here are 2 of the same ones we fished this year. So they're providing us as much additional information as if we would have fished, say, over here. And let's get the blue arrow so everyone can see. As if we fished like Yakutat or Prince William Sound or the western part of 3B. That would be something that would provide us more information, would allow us to reduce bias, potential for bias, and improve precision relative to fishing two areas that we've already fished the last 2 or 3 years. Those areas, though, outside of these, would come with additional costs, but that'll be an option presented to the commissioners. But benefits. As for 2B and 2C, we fished most of those areas in recent years. Certainly in the last 2 or 3 years, we've had very good coverage, and so the gaps here are of less of concern than they are in 3A and 3B and elsewhere. So regarding the changes in costs, or preliminary cost projections, they are counting continued increased costs and decreased catch rates that we observed in 2020. And they're still in the process of finalization. And these changes have further elevated the need for supplementary funding for the long-term viability of the FIS under all stock and market conditions. We're developing a design or have developed a design for country sampling in autumn this year. In comparison with summer samples, it's a top priority in the 5-year research plan and expected to generate some additional revenue to offset the losses incurred this year. So there'll be some actually additional slides after this, but we're asking you to note the paper and requesting that you provide scientific guidance to the commission regarding the design options to assist commission decision-making for the FIS in 2025 and subsequent years. But I will be bringing up something we talked about yesterday and bias and how with the renewed focus on bias instead of CVs, question came up, well, how quickly does the stock change over time? Ian in his analysis looked at changes of 15% over a 3-year period. Well, it turns out that changes are typically greater than that, and those changes are common across space, not just at the edges of the stock. So here we're looking at regions. These are 1-year changes in the mean O32 weight per unit by biological region over time, is estimated by the space-time model. And I put dashed lines at 15%. So this is a 1-year change. And you can see it's changes larger than 15% quite frequent in almost all biological regions, the exception being region 4, which is dominated by the flat waters of the Bering Sea covered by the Minstrel Survey. Yeah. This is 1 year. This is 1 year, not 3 year. So this is a smaller time scale. We've had periods like this period here where it was essentially a downward trajectory for a number of years from the high stock level, fish down to a low stock level, and then management came in and changed that. But the coastwide, the changes sort of average out, except in this very recently, that's 2022, 21 to 22. That was a very large drop. But we see frequent large changes at the level of biological region. If we go to regulatory area, some regulatory areas have even more frequent changes, either up or down at greater than 15%. If we look at 3 years, the timeline that Ian was looking at, then changes greater than 50% of the norm. In fact, 20% is not unusual and 30% is fairly frequent. Even in regions like region 3 here, we had the steady decline until about 2010 when it leveled out. But we've had more recent increases and more recent declines as well. Even region 4 has had frequent changes of that magnitude. So if we don't, and likewise for regulatory areas, so to state the obvious, if we don't sample, we don't see those changes. If we scale back sampling to say we're only sampling 20% of a regulatory area, that still leaves 80% where there could be large changes, and we're not going to see them. So I think there is great concern. The bias is a significant concern, not just in the long term, but certainly, if not if less so over a 1 year period, if we're doing designs like the reduced core design, whatever it was called, over like design 2024 design, and we sample that over 3 years, there's a lot we may be missing. And it shows that these figures show that it's more likely than not that we're missing some large changes in the stuff.
A lot of these, over time, the negative percent changes have a lot to do with weighted age. So is there a way to partition the change between abundance change and weighted age change? Because you can probably track weighted age change a little better. And
So we could do something similar on numbers per unit effort instead of weight per unit effort, and that would isolate out the weight change. Just focus on the numbers change.
Yeah.
These are most comparable to the commercial fishery, though. Our commercial CPUE is in terms of landed weight of legal size fish of 0.32. So this is the crossover to the commercial trends. But yeah, we could definitely do this in terms of numbers per unit effort. And in fact, actually, that's the way it's implemented in the assessment. The trend index is done in numbers so that we're not collecting numbers of fish, weighing them, converting them back to numbers in the model. We're just tracking the numbers directly and then we're tracking the average weight at age.
And those changes are probably less. That's exactly what you said. Yes, so I can work those up as well. But it's weight that's used for estimating stock distribution, right? Do we use all sizes or?
We do both, but I think that's an important point. Is that when historically when the commission has used stock distribution to inform distribution of quota, they have done it based on this index. So it's been integrating weight and numbers. It's unclear what they might use moving forward, but
Ray, it seems like you want to know two things. You know, one is the probability of different size changes in the different regions that you've got here, and then also the probability that you don't detect them.
Mhm.
Is that something that you can calculate? Because the big changes, yeah, they're really worrisome, but you probably detect them to some extent because of the correlations across regions.
No. For example, 4B. So the correlations don't extend that far. They're generally out to like maybe 3 stations and then it tapers off. So it's a relatively local correlation.
So dynamics are really
They're quite, the distribution is relatively patchy at that sort of scale. We don't have any maps on hand. So what's going on in Eastern 4A isn't really telling you anything about what's going on in 4B. What's going on in Western 4A might tell you a little bit about the very east of 4B. That's it. So we're not sampling 4B for 3 years. We basically lost all understanding of what's going on in 4B.
because the temporal correlation
There is
also decays.
Yeah. Well, there is temporal correlation, but it doesn't tell you whether if there's a big shift, it's still not going to capture it. It just says that the stations are moving together.
Okay,
Yeah.
so this really does answer that question.
I think so. It's-
None of these changes will be detected.
no.
and I think this really reinforces why we're sort of refocusing from CV to potential bias as the thing we're worried about.
Absolutely. Yeah, that makes a lot of sense for this. But what about like reduced designs where you do some sampling in these areas? Like
Right.
there, you've got a probability of detecting it.
I tried to work that out, although that was sort of how I was approaching bias in the past. Was the designs started losing control of what the designs end up looking like. But yeah, so if we're doing something like that 4B, 4A sampling in 2025, we haven't designed supplementary funding. Those are the densest parts of those regions. Not all of the dense part of those regions, but we're going to get a signal from them. It's certainly going to reduce the potential for bias in those regulatory areas relative to not something. But there's a lot that we're not looking at. And
A fundamental aspect of this problem that we have is that in many surveys, as you reduce station density, you just get sparser random samples. And so you don't have to worry about bias. You
Right.
just have to worry about precision.
This is the
But
downside.
for us, we're setting gear and then going back and hauling that gear. And so we need to have clusters of stations that can be efficiently fished, and the geographic distances we're working on. I mean, we span 5 different domestic surveys with this one survey. And so if we go to a sparse design, it's completely infeasible to do that. So we end up having to go to these block designs, which then leads us to this situation of bias. So it's quite different than a lot of surveys where you could just make things more sparse. And at least as it got imprecise, at least you could say, well, it should be unbiased, right? We can't get there. It's completely infeasible to sample as things get sparse.
One more survey. Last night you talked about some preference sampling ideas, these captain's choice stations.
Right,
Did that get
yes.
implemented or not? I didn't see much about it.
no, we're analyzing that data right now. So we did fish up to 1/3 of the stations were fished as called skipper stations or captain stations in 2B and 2C to help revenue. The big concern from everybody was that this would induce bias by having the preferential sampling of high density areas. And so I've done, so I started analyzing that, the data 2B, not onto 2C yet, and those concerns appear to be valid. You leave the stations, skipper stations out, you get a lower estimated index of density than skipper stations.
Well, sure, that's the whole idea, right?
Well, no, not necessarily. The whole idea was to make money, but the skippers turned out to be much better at finding the spots of high density, which sure they'll be happy to hear. Than I might have anticipated. One reason I thought that this might be okay for 1 year, one, we can always leave the stations out. But two, is it because of the correlation from 1 year to the next, if we're not fishing a low density station, we still had last year's value in 2B and 2C to influence that. We still do. But it turns out I thought the skippers are going to be fishing near other high density spots that we already know about. But they actually drilled in finer than that. And what they got was much higher than previously and pulled up those high density stations in their vicinity, the estimates for 2020. So we're only predicting at the grid. That's important to note. We're not getting an estimate at the location of the skipper station, which are all over the place. So we're still estimating at the grid, whether we sample the grid stations or not. When we have a skipper station nearby, it was so much higher that it would pull up the values for the nearby grid stations. So, and the bias is in the order of 10%. The difference, at least in the one variable that we did so far, is more than 10%. So, okay, we made some money, we learned something. We don't really want to do it again in a hurry.
Supplemented the biological data as well.
We supplemented the
You get
biological.
better length, age, size and age.
Yeah.
Do you think you're going to just throw those out then rather than doing some kind of reference sampling analysis?
So
I mean, it
I'm
sounds,
not so familiar. There's a type of analysis which we downweight them or something.
reference sampling. I mean, it's essentially estimating offset.
I know the name, but I know. Oh, okay. Yeah.
Jim Thorson's got a paper on it. That you basically estimate the degree to which skippers are better at selecting the spot than anybody else.
Kind
Is
of treated as a different survey.
Yeah.
Is it similar to how I treat, say, different baits or make a correction for different baits or different gear
I
types?
think it's slightly different because I think you can allow for the fact that those extra stations are actually preferentially better places to fish. There's one paper, because if you're using commercial data, you have to account for that. So I think that Jim was trying to come up with a method to allow it to be extended to use commercial data and then estimate the degree to which the commercial data would be biased relative to an fishery independent survey. It's maybe not 100% convincing that you can remove all that, but he did lay out a method.
One of the papers that I personally about the suggested doesn't work.
Yeah, it was ambiguous, I think, in the original one.
It's not necessarily. And you still have all the questions about whether you estimate a separate offset for each captain. Presumably they
So,
have differences, and very quickly or two, like the data just don't support that.
Yep. and aside from that, you don't have time. This time of the year, it's on the level of sophisticated modeling.
Yeah.
If this was something that was ongoing, we'd have no choice. If it's going to continue to raise questions in the minds of the stakeholders and others, it isn't.
Yeah.
I'll look into it just so I understand it. Anyone else's questions?
Is there any plan to have the full fish ever again? Or
We've never done the full design in 1 year. The most we've ever done was about 1,500 stations. But as Ray said, historically, we were able to do about 1,200 to 1,300 on average.
And they used,
So-
sorry, it used to be that we had something close to it, but with gaps. So from Logan will know all about this because he was in charge of the first back then. But from I think about 2000, 2001 to 2019, we had quite spatially extensive coverage in all IPHC regulatory areas. These stations out here were fished on an annual basis. These were new, but most of these stations were fished on an annual basis. Most of these were, and everything in the middle, except for some deep and shallow stations and some gaps. And the expansions we did from 2011 to 2019 filled in those gaps, added stations in deeper and shallower waters, and increased the footprint of the FIST by a third. But that third,
On a roving basis. You
Yeah.
couldn't do them all every year.
we didn't do them all at once because it was too much. But we did a chunk, one regulatory or two regulatory areas each year. So we did have a FIST that was annual FIST of 1,200 something stations, but it had gaps. But the gaps were not like this. They weren't generally big blocks, except for maybe a fishing channel here or Bowers Ridge fishing channel, Prince William Sound, or Bowers Ridge was never fished until 2017. Northern California was not fished until 2013. But for the most part, the coverage was spatially extensive. So that's the closest we've had to what you're talking about fishing before. This footprint, we haven't been able to do that since. So if we do it, it's going to be something like the base block design. We do it over 2 to 3, or in some cases 5.
So starting design for 2025. What are the options?
So one thing is, okay, so I mentioned that there are charter regions that would give us more information than the ones we currently have in this design. So this one way of thinking of this is the most fiscally viable design if we're sampling every biological region. But we're going to get more information in 3 and 3B by fishing charter regions that aren't these two. They would come with increased costs. Is that data, is that increased cost worth the benefit? I guess is the question you might consider. That we would, the scientific benefit we would get from fishing areas that we haven't fished for a couple of years. Another thing I've been considering is we're going to fish designs like these where we have contiguous adjacent charter regions, more appealing for bids than if we have one charter region here and one charter region up here. We can get a bid on 2 adjacent charter regions as a single bid. So we try to do that. Haven't done it here. So if we were to put in an alternative design, it would be maybe a charter region here and the adjacent one, or I better remember to use the blue arrow. 2 charter regions in Western 3B, or 2 charter regions in Central 3A. We could reduce the loss in those charter regions by thinning the number of stations. Now, we've moved away from that. Blocks are more efficient, but in the case where you're losing money you can still reduce the amount of money you're losing by reducing the number of stations to some degree. There'll be some increased running time, but there's still less time on the water and less maintenance. And while getting essentially, if it's done in a randomized way, we don't need all the stations in each block to get precise information for that block. That's a lot of data. We do it because it's more efficient for fishing. But if we're losing money, we can reduce the losses to some degree by reducing the number of stations. So if we were to move, say, these two and fish these, we went to fish those ones and we fish those ones instead, there may be a more efficient way to do it, to make them more efficient to commissioners anyway. Otherwise, I don't know anything else that's viable in 2025 in the absence of some surprising supplementary funding.
A charter region is something that can be fished within like what, how are those defined? Like similar to what you have here, right?
Yeah, they're typically blocks of 50 to some cases 90 in Charlotte stations. Then I guess have been developed over the years as they're large enough that they're appealing to charter bidders.
Yeah, it's a way to chunk it up for a bid entity so that people can bid on this region. And it's traditionally been 3 to 4 weeks of work. That's how it's been designed. And so they know that, okay, if I win 2 charter regions, I've got, 7 weeks of work. They'll often bid them adjoining because there's economy of scale. They can cross border fish. They can fish one way to a port, fish the way back to the port they started on. There's a variety of benefits for chunking it up in kind of bite-sized pieces. And we just call those regions, but they're traditionally we had the whole coast mapped and we just chunked it up into 27 regions, but the definition of a region has morphed a bit as we've added deeper stations, done block designs, things like that. We've had to chunk them up differently for contract purposes. But
something like this, which is 60 stations, we tried to get that fished a couple of years ago as 30 stations, and we did. That just wasn't a viable postage for getting on this. 60 stations is a good, seems to be a good guess or value. Likewise, down here in 2A, we consider this as one charter region of 60 stations as well. Yeah, so even though that's not what, as Claude mentioned, this is not what we traditionally would have thought of as a charter region, which did not cross regulatory boundaries. This works as one region that be fished by one vessel in 3 or 4 weeks.
I remember last year we talked about snap gear and any updates.
So we used it in 2023, but I don't think anyone did snap.
We didn't get any bids last year.
Yeah.
It's not proven to open up the bidding pool the way we thought it would. We thought there were a bunch of quotes that would join the party with snap gear, but we've sort of confounded because we've opened it up to snap gear at the same time that most of these vessels have been swamped with sablefish quota. As opposed to really just struggling to catch all their quota, their normal quota in addition, even before they think about extra quota. So it's hard to say. We still have snap gear is still an option, and we can continue to accommodate vessels. So we're not going to. We're going to. It's potentially in the long term still going to create a larger pool. The idea would be that we could bring in new vessels and that maybe we could make the bidding a little more competitive, but hasn't really proven out to be a huge difference.
Is the industry happy with this program? Do they see this as a nice opportunity sometimes, or are they frustrated working with it? Conversations about that. I wonder if there's things that like, I don't even bother with it anymore. Like it takes forever to come checks or something like that.
Mixed.
Next.
I mean, I think that some of the boats have figured out how to integrate this into their business plan, and it works for them. In the last few years, again, because of such large sablefish quotas, it's maybe not as appealing to some other vessels. We certainly have vessels in the fleet that are that make more per day doing their own fishing than they could on our survey. And we've never been able to, rarely have ever been able to entice them to bid on our survey. But
I think we've seen a graying of the fleet over the last 15, 20 years as well, where people are starting to get out of the industry. And as they get out, those boats, as they're picked up by other operators, they're often not maintaining their bids. So that's not something that they continue to do in a lot of cases. So we see attrition that way as well. We thought introducing a snap option would help offset that to some degree. And, and maybe it's a victim of the sablefish abundance right now. I don't know.
Have you done a survey at any point asking people why they don't participate?
I'm not sure how that's dealt with today.
we have.
Seems like it might be-
We're in contact with all the various fishermen's associations. People know about the opportunity by and large, although they may not know the details. It's also,
like I was going to say, we have our research advisory board, which meets in November, and we have people that are board members on that the vessel has previously bid, but now it's owned, it's being run by a different person. And they'll come to the RAB and they'll be like, oh, well, how come you're not fishing in here? But then we keep telling them like, oh, it looks like the commission is going to approve a bid in 4A, but nobody will bid.
and
Right.
honestly, the uncertainty in the design in the last few years has led some vessels to have less interest as well.
Yeah.
Because they used to be, they could sort of count on, well, I'm pretty sure if I put competitive bids in, I could get a couple charter regions. And now we don't really know what the next year's design is. So I think we've had, that's caused some challenges as well. It's not as stable an opportunity as it was even 3 or 4 years ago.
We went through a period of time where we would do multi-year contracts. And so typically they were like a 3-year contract, the ability to break the contract each year at the end of the contract. And typically those, they'd estimate what their cost inflation was going to be over the 3 years, and then they would bid it, we'd award it. And typically probably 75% of those, they would last the first year and second year. And they would break it off for the third year and rebid it. But we had a lot more people bidding at the time. And, they would take advantage of the averaging it over those 3 years. They were making extra bank year 1. They were making estimated bank in year 2. And then they were throwing the third year out on a gamble to rebid it. Right. So you get that sort of dynamic happening with that. But that was with stability in station design, et cetera. We don't have that right now.
Yeah, it might be worth thinking about a survey to figure out whether there are some simple things you could do that you get more people involved. And then you've got anecdotal information about it, but how many people does each one of those factors represent? It might be, integrating stability would give you 10%, but if you could do something else, that would bring you 50%. Or it might be just that everybody's making money on sablefish. There's nothing you can do at all. Survey could tell you. You have a price. The price of the fish is something you can't control, but there are some levers that you might still be able to tweak. Apparently, Snapgear was not one of the ones that had much influence, but I think it's still worth thinking about.
We produced an internal briefing note last year that had more than 20 different options for trying to make this more efficient. And we went through it with the commissioners and to some degree with our RAB. And we've just been working our way down the list, trying all the things that we thought could possibly either reduce bid costs or increase revenue or increase efficiency. But we're sort of running out of
Yeah,
other efficiencies to try.
I mean, skipper stations work great. They give us money, but some they unfortunately didn't pay enough. Some prices came down even to Pink, and then Pink didn't touch as much as Chum. 20% less.
A lot cheaper than I got.
Yeah, exactly.
So we have, so they want us to comment on the scientific
I mean,
about this.
I think from the comments yesterday, I guess commissioners would like to know if we kept doing something like this for 3 years, how what are the scientific concerns, or what are the scientific issues with fishing something that looks like this, or even this? 2024 for another 2 or 3 years. And bearing in mind, we've already had scale back designs for 2 years. We're talking about 3 more years ahead. That's really getting into 5 years in some places. More in areas that will be really not sampling. So it's not just.
So this is even more extreme than reduced core. Is that right? Or is this reduced core?
No, this is more extreme than the reduced core in the core. This is like a reduced core.
This is a different design.
It's a different design. So we're sampling less in the core. The reduced core would have at least 2 charter regions feed fished in each of 3A and 3B. And I think 2 and 2B and maybe 1 and 2. I can't remember exactly. But it wouldn't, the reduced core, it's a core design. It wouldn't have anything in 4B and 4A or 2A. So there are pluses to this. We're getting some sampling at the ends of the stock relative to the reduced core. We've got less sampling in the core itself.
So we can't extrapolate off from table 2 with the neighbor. We can't extrapolate off reduced core to say the CVs are going to be even higher than reduced core. Because we might actually be comparable.
We probably could, yeah, the first one level would probably be at for 4A, 4B, and 2A, they're going to be there. 3A and 3B, they're going to be. But in terms of bias, we're winning in places, and again, it's the same thing. Reducing bias from the ends and stuff, reducing possible bias. We're getting.
This is one of the challenges we're in, which is that it takes us weeks to set up and do these simulations, either in the space-time model or in the MSC or in the assessment. But yet the commission is wanting to have this sort of plug and play options, and we just can't preload enough information to be able to tell them exactly what happens under each, adding and subtracting individual charter regions. We can give them a sense, and that's what we've tried to do.
Yeah.
Try to come up with this kind of scaling of designs to give them a ballpark and say, okay, on the spectrum, here are 3 points we've tested, and here's kind of where you are on the spectrum with the options you're choosing. But I don't foresee a time when we can really do a real-time cost-benefit analysis for them, because there's just too many mix and match options for us to do that. And it all takes a long time to set up and simulate, because it has to simulate through the space-time model, all the way through these pieces, which basically it's all the 3 of us did this summer, was simulate these 3 designs.
I imagine that just reflects like dissatisfaction with the options in front of them, right? Like, you want to tweak it when you're
And uncertainty about funding. I mean, we really didn't know that the 2A and 4A and 4B options were on the table until the work meeting, which was 2 weeks ago. So that was new.
Yeah.
It's good news
Yes.
that we could get some additional sampling there, but if we hadn't been able to.
Incorporate that into the other thing we're having, we talked a lot about bias. It's not always easy to communicate bias and to commissioners, the importance of that, or understanding the potential for it, because you can't quantify it in the same way that we can CV. CV is a number. They now are kind of familiar how it works. But we say, well, we can't say there's going to be a 20% bias. We never know. So I talk in terms of increased potential for bias or high chance of bias. And I think those new figures are going to help communicate that more effectively. So hopefully, we're writing a briefing note to commissioners right now that's going to address some of these issues, communicate some things. Bias is one and it's going to struggle getting across. glad that we're shifting focus over just precision, they got you got stuck. Because it was easy to communicate.
And so now that you've done the analysis showing that 15% is probably overestimated, especially for the 3-year.
Conservative.
Yeah, that's sort of underestimated. Are you thinking that you'll redo some of the analysis you did with the stock assessment? Is the 15, or is that too much work to do?
We could, but honestly, I think our time is better spent on other things.
Especially with the assessment coming up.
Yeah, I mean, I certainly can't do it this fall. And whether that's really worth, displacing a couple of weeks of development time on the next full assessment, honestly, I don't think it is. It's still all predicated on how big we think the bias might be. I think we've demonstrated that things move in the direction we would expect. If we fail to detect an increasing trend, there's temporarily some fish left on the table. And if we fail to detect a decreasing trend, we potentially are harvesting more than we think we would. That's not completely offset by the fishery data in the model, and it's going to compound over time. The longer we go, the worse that pattern get. Whether it's a 1% or a 3% change in SPR, I don't know that really provides them.
That doesn't provide them anything, but translating that to dollars, I think does. That's a threefold difference in dollars, right? So that, I mean, to some extent, I see that analysis that you did as a really valuable communication. Loss of precision and increased potential for bias translates into dollar costs. And I think at this point, since it's done for a value that 15% trend, which is an underestimate, then when you translate that into dollar costs, that's also an underestimate of the potential cost of survey as fully as planned. I understand that doing it again is additional work, and maybe there are other things that you'd like to do with that. But communicating an underestimate of the true cost of the survey seems not terribly useful.
Yeah, it's a challenge.
I mean, Ray, could you go back to that 3-year figure that you had we got the 15% essentially by eyeballing this figure and saying at the coastwide level, 15% isn't that far off. And yeah, there are bigger values and smaller values. I don't know that we might have even actually just taken a raw average of the values to get to 15%. I don't recall exactly. And then we rounded it to the nearest 5% or something. But that's sort of how we got to that 15% at the coastwide level.
Yeah, that's not terrible at the coastwide level.
It's not that bad. But I think what's come up during this meeting is that if you have interest in the trends and dynamics within the biological regions, things can be a lot different than 15%.
Yeah, for sure.
Part of the reason the coastline is so because other regions can't each other out. Some go up, some go down. But
Right.
Yep.
Regional changes large.
I mean, honestly, I think from our discussion yesterday, understanding whether we should or shouldn't expect the fishery to continue to provide us with a non-hyperstable index might be more important than a small refinement to the simulation we've done. I guess I've got my fingers crossed, thinking, well, maybe we're going to get some supplementary funding and the pressure will come down a little bit on this, because we'll have enough that we won't have a design that's clearly putting us in the real danger zone for large amount of bias. But we may well have several more years still of reduced designs.
Well, I think that maybe that's something that we can just note so that it gets up to the commission level that these have real costs. Maybe you can put in some of the numbers from there.
Sure. And I didn't include them in the PowerPoint, but those figures that I quoted out of my notebook are in the document. So those details are there.
We might just put some placeholders in there. So
Sure, we can copy them over.
make sure we get that right.
And the one, I think that from our discussion, we had talked about when we first got them, we were looking at the case where the stock is increasing and we don't catch the fish. We were thinking, well, it's not really fair to say the foregone yield isn't lost, and some of that's going to be there in the future. But I think Sean's point about the asymmetric relationship, it's really the downward case that matters the most, where the stock is going down and we don't know it, and so we harvest more than we thought, and then you can't get those fish back. You have to pull back more down the road to get back to where you want it to be. So I think that's probably the one that's most important to highlight for the commission. Although in the long term, I do think it's important that Alan was showed with the MSE that
exactly.
even in both directions, there's still some foregone.
And I think we brought this up before, but there's no appetite for something like surveys every other year. Like if funding is external and you do one every other year, you're kind of spreading costs out. Whereas if it's self-funding like this, the problem's always going to be there because until prices change or situations change.
Well, we could do a larger survey and then a smaller survey, sort of alternating. But we lose less money by doing a small survey in money-making areas than we do by doing nothing at all, because we still have all the fixed costs associated with the survey. So the best case, I think maybe the best in that scenario would be maximizing external funding by doing a bigger survey and then a smaller, closer to net revenue neutral, and then back again. And these things can change quickly. I mean, we went from really a pretty complete survey that paid for itself plus just a few years ago, post-COVID, with a little drop in price and catch rate, and then all of a sudden the ledger's in the wrong direction. But a little bit of change in catch rate or price, and we could easily be back to where we can do a much broader survey. Which is actually something we've been having a challenge conveying to the commission, which is that it's not about sort of sharpening our pencils and figuring out how to do this better. It's these things we don't have control over, price and catch rate. And so these things are inherently going to change. There's likely to be a wide range of potential for net profit versus net loss that we just never going to be able to control, even if we had great way to predict it.
And you see that even with the supplementary funding for next year in 2A and for A and B. The supplementary funding is to the dollar amount that we put a pin on and said, well, this is what we think we're going to lose based on these amount of variables, but there's a large amount of uncertainty there. And so there's still a failure to understand that, look, you give us $137,000, you say that that's the middle number that we can do surveys in 2A, we still may lose money. So it's just going to compound year on year. But yeah, I don't think there's an understanding of the kind of.
Another issue with biennial surveys is the variability in the population. We saw that in that 1 year change in the coastwide. I mean, it changed 20% from 1 year to the next at the coastwide level. And when you start missing those sorts of things, then you really get behind the ball. We've seen that with other stocks.
Did the stock actually change by 20%? But you should also look at actual changes in the model. That's more of an indicator of what the stock might do.
I mean, the
We certainly
demographics
smooth those out. It's interesting though that at the coastwide level, we're certainly smoothing out some of that up and down with the stock assessment estimate. But at the region and particularly at the individual IPHC regulatory area level, a lot of that change is real. Those fish may be in one area 1 year and in a different area the next year. And so, and because we've had cases where we've documented a very large change with a very precise survey, and 3B comes to mind. We had a very precise survey 2 years in a row, and there's a large change in abundance. The conclusion was those fish were passing through. We caught them 1 year, and the next year they weren't there. They were somewhere else. And so, not so much at the coastwide level, but definitely in terms of stock distribution, a lot of these changes are real. The stock really is highly variable across its its geography. As year classes come through, each year class has its own distribution. And there are some patterns. They tend to fill in from the ends of the stock back to the core. But it's very much, each cohort has its own pattern of how that works.
Makes sense with spawning spread over the region, right? You're not gonna hit equally in terms of spawning success or urban success in all regions.
Exactly. The 87 year class, which was the biggest one in the time series, really showed up in western areas. And with that, a lot of the trend you see here in the declines in some of the western areas are largely attributable to the decline in that 87 cohort, decline and movement of that cohort over its lifetime, farther east. And as we caught those fish out, we caught over 220 million pounds out of that one cohort for its lifespan. It's a huge cohort.
The issue too with the biennial is when, if you did catch, the survey saw this huge decline, there were a lot of questions about it or an increase or whatever it was. You have to wait 2 years for that question to be answered. And sometimes that can cause issues even in the stock assessment.
Yeah, it seems like that's not.
And as we've noted, the industry and the commissioners do look at these data, these results, and helping them form not only decisions, but their internal opinions and understanding of the stock as well.
As you said, there's no sharp evidence.
So they try to make separate inferences from what all the scientific analyses say based on data alone.
I would say industry, Ian gave the example of the big change in 3B, for example. And a lot of people actually said, oh, on the water, they're like, oh, we're not seeing the catch rates we were expecting, blah, blah, blah. You said there was a lot of fish in 3B and they're going to be coming through, but we didn't see that this year. And then the survey results come out and they match that observation. And then people feel, oh, okay, that's great. Sometimes it doesn't happen that way. They want to understand it more. They're comparing it to what they saw in the water, and then we'll go in and dial in on their areas, with the different explorers we have.
I'm thinking for like decision making.
I think the decision making comes in a lot in how they are allocating the stock across regulatory areas, even though they don't want to use stock distribution from the survey in a formal manner. I think they are using it behind the scenes to justify some of the decisions they're making.
And we did for a period, more than a decade, we had a fairly formulaic approach, which was to use the stock distribution based on 0.32 catch per unit effort. And that was a pretty close guide to how the mortality limits were going to be distributed. And that was the status quo for, like I said, more than a decade until we got to about 2019, when there was an agreement made for 2A and 2B. And then Alaska still largely based their distribution off this. And then in recent years, it's become a little harder to classify.
And Alaskan stakeholders are interested in using the distribution just within Alaskan areas to help make decisions about how to allocate it only among Alaskan areas, excluding 2B and 2A. They've been having some discussions about that. But there's other factors that are influencing the allocation, like the amount of quota in 4C, D, and E that, with various things in recent years, the quota's been at lower levels where they're saying, well, we won't even be able to open our processing facilities, or we wouldn't have a fishery. We need just another 200,000 pounds. And so that's the decisions the commissioners are making that affect them departing from these distribution.
Is there a way, if you made some, in the short term anyway, run some scenarios of prices, you could include the deficit, potential deficit in the MSE simulations, couldn't you?
The deficit being
For when you're simulating, because you're simulating different design outcomes. So you're projecting the CPUE on the survey. I wonder if there's a way to tie that to come up with potential revenue in the survey. And
Yeah.
it's like we're being asked to judge these things. It's basically a one-off kind of thing. It's the same issue, right? It's,
I mean, the MSC is really focused on projecting long-term. How different would it be just developing a model to project survey CPUE for the next 3 years? But if
well, you're going to do that anyway for exceptional circumstances.
Yep.
So you presumably could.
Yeah, I mean, it's
you're given a
definitely,
price scenario that you could put the expected survey cost.
yeah, it's definitely an output.
Given some price assumption for the
Yeah,
short
and I
term.
mean, I think that's part of what Ian was saying. There's so much variability, and there would be quite a bit of variability from the MSE simulations in the catch rates from the survey, because there's a lot of variability there. There'd be the uncertainty in the price.
They just make the same assumption. Even if we know that there are other factors that influence it, but we know that the inverse demand should hold in equilibrium.
But how different would the using the MSE be than the tool that you guys have already created? Would it have a different, because you're providing an uncertainty with that tool.
Well, one of the things is like Ian's foregone catch.
Yep.
It would be made up if you were making consistent decisions. You would make it up.
Okay.
Just be another way to come up with an economic impact.
I think the way to do that too is to states honor that constant. Then you can compare scenarios with the same level of risk.
Okay.
Once you've got,
Yeah.
changes in risk, changes in revenue.
Yeah.
Yeah, that's apples to oranges.
It's another solving loop for projection.
Yeah, that's what I was just trying to figure out. Want to
That's one of the
stimulate.
things that happens, right, with these poor survey designs is you want to maintain the same probability of bad things happening, you have to back off more, right? Because you're flying blind essentially.
I think that is actually fundamentally what the commission was asking you guys. Is that the case? Is there evidence based on the work that we've done that they would need to do that under these circumstances? I think we produced that evidence at 3 different levels. It maybe not be quantified exactly to every design option that we have, but the general trend is pretty clear that there is going to be additional risk if the stock is. There is potential for the stock to go down and we don't detect it. And if that's occurring, it will propagate into the assessment and turn into higher than expected fishing intensity and lower than expected biomass. And it also translates into long-term performance loss in the MSC.
Yeah, I mean, these are some of the things where you could say, the team might say, this is going to get worse. Let's say something like decision-making. Right? We don't. It's something. The decisions aren't anchored on something. Versus doing it, you may actually reduce the amount of loss you're going to incur over the next 3-4 years that you have to ask for supplemental funding. And it keeps coming back to, if you want to ask anything to do with risk, you need to define it and stick to it. But we have to be committed to a certain The other thing is like some of this is also conditioned because if the stock is in some reasonably healthy state, who cares if the probability declines higher?
Well, and
Still
likely
absolutely low.
at a higher catch rate, we're likely to be able to have a larger survey that's revenue neutral.
Right, so it switches it to, why keep driving this stock down? For economic reasons, like you're just gonna keep the headaches continuing.
I mean, maybe that's something that could be explored at some point in the see. I think it's really pernicious feedback there, whereas the stock declines, it's more important to have greater precision. And then it can be on the assessment because whereas at high abundance, you get a lot of room for making mistakes to catch it later. But as the stock declines, your information content gets worse because the survey pays for itself. Its catch rates go down.
Yeah, and then you're on top of that compounded with decision-making variability. It's an acceleration. You don't even have to, it might not cause a stock collapse. That may be not what you're talking about, but it's an economic, really bad economic situation. Especially the fishery doesn't seem to like the catch rates where they're at.
Currently built into the NC, right? Sort of uncertainty in the assessment going up as the stock size declines. There's no mechanism for that. No, but in the simulations of the core and the reduced core designs, we had that the bias would be greater for the survey and the assessment when the stock was lower, because when the stock was lower, we're going to a base block every 5th year. And when the stock was higher, we're going to a base block every 3rd year, which sort of reduces the bias. Oh, okay. Yeah. So we captured there. Yeah, I mean, in a very archaic way, it's not, but we did realize that could be the case. And so we wanted to have a little bit of that in there. That's easy to change if we wanted to investigate. What is the real effect of that or something? But it's only on the bias, it's not on the CVs. You could easily implement something on the CVs as well.
It'd be interesting to see, like if you showed, let's say in the 3 to 5 year window, the situation remains the same with the prices. You projected out, I'm talking accumulated losses here, the need for external funding of $8 million. Then the conversation about something like close-kin market capture, which initially was like, it's super expensive. It's over $1 million to get the base. That's starting to look like maybe a good investment. When you can't afford to do a survey, you still would be getting information about abundance from that kind of thing. Because you're sampling the catch. Sense of abundance that way. So look, just stepping, a little bit higher level thing to see.
We'd be all in if we gave up the survey and went all in on close can. That would be.
It could be an alternate, could be a solution, right?
No, I know, but it would certainly be a commitment in
It would take a bunch of money. I mean, there's no doubt you can do it. You take a bunch of money, but maybe that money is not ridiculous
compared to
how much you're going to have to need anyway, and not be any better off. You're still going to be subject to price volatility. That might actually be a useful application of the MSE without having to, yes, it's contingent on them making consistent decisions, but big picture, it's probably going to be robust to their decision-making variability. It's 5 million, maybe it's 8 million. Still, it's a lot of money.
Is there any relationship between the TCU price?
It's not straightforward inverse relationship. It's very likely compounded with the price of blackout because of such it was I tried to build a personal model. It's just but that's of course like that's very those multiple additional factors, but something like see inflation, but because of that, on average, this would be. Consider specific carbon inflation.
Sure, but as you said, the blackout, there's no point in considering.
Yeah, but you
isolation.
can assume the blackout is constant.
Yeah.
The rise of Atlantic halibut is throwing a wrench in that relationship as well.
Oh, interesting.
That fishery is now a third, I think, the size of the Pacific halibut fishery.
And those are completely substituted.
Apparently. And that fishery is producing fish all year round, even during the winter. The fishery is closed.
We've got some good notes to call out in the document. I don't think we've got like good next research direction. So it's not like it seems like you guys will run this to ground. Well, it's large impacts.
Did we wrap up? It's not now.
Yes.
Okay. I suggest we break for lunch then, so we have presentation and some more items to discuss. 1:30.
Thank you.
Thank you.